Hello all, welcome to this week's session on this algorithmic thinking in bioinformatics course. We are going to talk about an important problem in next generation sequencing. So these latest generation of sequencing technologies and the reads that it generates on different individuals and patient samples. And we are going to see how we can apply algorithms to those measurements that are coming out of these sequencing instruments in very large numbers to locate disease-causing mutations. And the heart of these analysis are really string matching algorithms or pattern matching algorithms, and that's what we are going to look at today. So welcome to this session, and let's get going. In terms of the context seen so far, so this is the same context that we have been having for every topic. So after we have introduced and motivated the course, we have now looked at a handful of problems, handful of biological questions and associated computational problems, right? And then how we can develop algorithmic solutions for these problems, so that really bring the power of computation to address these biological questions by analyzing the large amount of data that comes out of these high throughput experiments, and specifically these next generation sequencing experiments, right? From these next generation sequencing instruments, the large number of DNA sequences that are read by these instruments, which we also, you should know by now that we are calling them reads, right? A read is simply a sequence of DNA that is read by a sequencing instrument. Today we will think about this question of how do we identify disease-causing mutations in a particular patient's sample, right? And then these sequencing instruments will generate numerous amount of reads, billions of reads from this patient samples, right? Or millions or billions of reads from these samples. And we will have to identify disease-causing mutations, but now we don't have to do it from scratch. Unlike in the problem of assembly of genomes, right? In the genome assembly problem where we had to build a large graph, the De Bruijn graph, and find Eulerian paths in that graph. Here we have a simpler problem, which is that we are already, someone has given us the reference genome. So we already know from the Human Genome Project how the human genome looks like. And then we now have another human, a particular patient, let's say, who has an ailment, and then we do not know what mutations, if any, are causing that disease. And we would like to compare the reads from that patient to the reference genome to see what is different between this patient's genome and the reference genome, and identify such differences, which are these mutations, right? So how do we take this ambiguously defined problem? I have just hand waved now on what the problem is. So you have the reference genome, which we have already constructed, and then we have this set of measurements from this new patient, and we want to compare the reference genome to the patient's genome and identify where the differences are, right? How do we formulate a concrete computational problem around it, right? And how do we figure out algorithms to solve this problem? Okay, that would be the focus of this session. And to be a bit more specific, what we have done so far is reconstructing this reference genome from billions of reads. So that is done, right? So this is what we had done by considering this De Bruijn graph, and finding an Eulerian path in this De Bruijn graph, right? So this will convert a set of reads, numerous amount of reads from a particular individual into a reference genome, right? So this reference genome is a representative genome of the whole population of individuals. In the case of the humans, this reference genome would be roughly 3 billion letters, of ACGT spread across all our chromosomes, right? And 20 plus pairs of chromosomes, right? So we have this reference genome, but this reference genome is only a representative genome. Your genome and the reference genome, or my genome and the reference genome, would be mostly identical, but every 1 in 1,000 roughly, right? Or about, so every 1 in 1,000 roughly, or which amounts to about 3 million, right? in 3 million locations, the reference genome and my genome will be different, right? Similarly, in about 3 million locations, the reference genome and your genome would be different on average, right? So every 1 in 1,000 letters would be different. And how do we figure out what that is, right? I mean, there are two ways to go about it. I can get all the reads from my genome, and I can again construct a De Bruijn graph and then find an Eulerian path in the graph, right? That is going back to the basics. So you have you run the same algorithm that you use to build your reference genome de novo, right? So, or from scratch. Or you take, if you want to know your your genome, you will take all the reads from yourself. Then you construct the De Bruijn graph and find the Eulerian path to figure out what your genome is, right? So any individual's genome can be figured out this way, right? We can construct the genome from scratch using all the reads. But that would really not be standing on the shoulders of giants, so to speak, right? Because we have this reference genome with us, and we know that except for this minute fraction, which is like 0.1 percentage, right? So 1 in 1,000 is really 0.1 percentage, right? Except for 0.1 percentage, 99.9 percentage of the reference genome and our genome are going to be the same, right? So why go through the whole process of generating billions of reads and then constructing the reference genome through these Eulerian graphs? Constructing our genome using these Eulerian graphs. Can we start with the reference genome and then map our reads, which is the reads from a particular patient, to the reference genome to figure out where we differ, right? So that is what the strategy we are going to follow, and that is what the strategy that the field and the community has also followed, right? We will start with this reference genome that is given to us. That is a very high quality reference genome because multiple groups have worked on it, and they keep on updating and refining the genome. So even very recently, right? So there was this project called telomere to telomere sequencing, and so some of the repeat genomes were not properly sequenced when the first reference genome was published. Human reference genome was published in the 90s, but now recently we have more accurate reference genome that also covers some of the repetitive regions near telomeres, which are some regions of the chromosomes, right, ends of the chromosomes where there are more repeats that occur. And we have seen that your De Bruijn graph will become a bit more complicated if there are many repeats, and you won't have a unique reconstruction of the genome if you have many repeats in your genome, right? But those repeats and other issues have been addressed over the years, and we now have a very high quality reference genome, human reference genome. So we have this reference genome with us, and we would like to utilize this high quality reference genome and map the reads, the billions of reads, from a particular individual sample, right? And this read, as I have said, is one read is simply a sequence of measurements, maybe ACC, GTTT. This could be one read that comes out of a sequencing machine, right? This is just basically a fragment of DNA that has been taken from myself. Let's say if I'm doing my genome, a fragment of, all the DNA from my, extracted from my blood could be all chopped up into pieces, and then each of those DNA fragments or pieces are now fed into the sequencing machine, and then that reads out what the DNA is, right? And so each of those will be a read. A read is just a very short DNA sequence, and the letters that make up that DNA sequence that is output by the sequencing instrument, right? And you have many such reads from all the DNA fragments that are collected from a patient. Now, how do we map these reads to the reference genome? Meaning that figuring out which location in the reference genome these reads have come from, right? And we would like to map these efficiently and confidently. Efficiently in terms of running time and the space utilized by the computer that is doing this mapping process, and confidently in the sense of if the reads, if we map it to a particular location in the genome, we are confident that that read came from that location and not any other location. Okay. If we can do this process, if we are successful, if we succeed in this, then you can identify the mutations in this individual, right? So that's the strategy. Okay, I hope the context is clearer now. We start with the reference genome and we have lots of reads. We want to map the reads and so that we can identify the disease causing mutations in an individual. So let's just to make sure that it is even clearer, let's just put it in pictures. We call this the short read mapping problem, right? So mapping numerous short reads to a large genome. And so the large genome is this. In the human case, this is this 3 billion letters. Of A, C, G, and T's, right? And the short reads are these numerous short reads that come from the copies of this, right? So you may remember this. So this is the reference genome. High quality, meaning the amount of sequencing errors is very less and the repetitive regions are properly mapped, and so you know what are the letters that make up this reference genome, right? And then you have your patient sample, a particular individual sample. You start with the DNA extracted from that sample, the genome extracted from that patient sample. You do certain experimental procedures to amplify this DNA multiple times. So you make copies of this DNA, right? All of these are the same patient's DNA. So all of these are copies of the same patient sample, right? So copies of the same DNA molecule, right? And we know that the current sequencing technologies cannot read all the way from the whole 3 billion letters. So you randomly make some breaks in these. Using certain enzymes that will break DNAs into shorter fragments. So all of these copies of the same DNA from this particular patient are being fragmented into these shorter fragments. Now each of those fragments, so these fragments, this, and so on, right? All of these fragments are all fed into the sequencing machine, and then you are able to read them. So maybe this one will read AACC, this may read GGATC. The reference genome may have GGATT, but this particular patient may have GGATC. Okay, the C will be a mutation, right? But this AACC is exactly matching this AACC here. Okay, and then here again this may be CCGGA. So this is an overlapping problem. You have this CCGGA here, and similarly, right? So all of these are these short substrings of this patient's genome, right? That is going to be the reads that are output by this machine. So now you have multitude of these reads, overlapping reads, short fragments, short reads. That's why we call them. While the whole genome is 3 billion letters, the reads may be, each of these reads may be anywhere from, let's say, 25 to 200 base pairs. Right? They are really small compared to the whole length of the genome, which is 3 billion letters. But on the other hand, there are millions to billions of these reads. Right, so you have millions to billions of these reads, and you want to be able to efficiently map them to the genome. Okay, because the genome itself is a billion letters, you want to make sure that every bit matters, right? So every bit in your memory matters, and then because the reads are also counting from millions to billions, you want to make sure that each read is assigned to the correct location in the genome properly, right? So there may be some gene here, I mean some read here. You need to figure out that read comes from this particular location of the genome, right, that maps to this particular location of this genome, either exactly or with some small mismatches, which are the mutations in the particular patient, right? So for each of these reads, you need to figure out where in the genome it comes from by scanning through the whole genome or using some efficient procedure. Okay, that is the problem in pictures. And I hopefully, when I conveyed the problem in pictures, I have told you these, implicitly said about these challenges in the short read mapping problem. There are two types of challenges. One is computational, right? We want time and space efficient mapping because the number of reads are also in the billions, and the reference genome is also in the billions in length, right? So it is a very practical matter that if this technology ever were to be used in the clinic for finding disease causing mutations, we need to have very efficient solutions, right? Both time and space efficient, so that if you can think about it, so if your solutions are not time and space efficient, then you need to have these large workstations that can only do the processing and identify these mutations. But if you can run your computations on a laptop or on a mobile phone, right? So if you have such efficient solutions that you can just run on your laptop, then it's going to immediately extend the usefulness of this in a clinical setting, right? So computational challenges and solving them in a very efficient manner, time and space efficient manner, would immediately translate to larger clinical utility of this technology of finding disease causing mutations. And the other one is statistical challenges. We are trying to assign these reads to when these reads are all mixed up and they are all fragmented and then they are coming out of the sequencing machine, we lose the identity of which part of the genome they are coming from. And that is what we are doing in this read mapping problem, that this comes from this middle of the genome, this comes from more like the end of the genome, and for every read we need to know exactly where it is coming from, right? But if some of these reads are coming from repetitive regions, right, what I mean by that is, let's say that there is AA, CC, GG, ATT in the beginning of the genome, and also somewhere in the middle of the genome also you have AA, CC, GG, right? So then if you see this read AACC, is it coming from the first location here, or is it coming from the second location here? So is it this or this? Is it coming from this location, which is the first location, or is it coming from this middle location? Where is this read AACC coming from, right? So that is a question mark, right? We may not be able to accurately identify the location of a read such as AACC, which comes from repetitive regions, meaning that they have certain letters that are appearing in multiple places in the genome, right? Having longer reads, so smaller reads may have more issues with this repetition, but if you have longer reads like 200 base pairs, then having a long stretch of 200 nucleotides in multiple places in the genome will be rare, right? So larger reads will partially solve this problem, but still there will be some issues where certain reads will come from repetitive locations and we won't be able to confidently know where it is coming from. Okay, so then the question is, can you give some continuous quantitative scores to confidently map those reads to the correct location? And sequencing errors can also exacerbate this issue, can make this issue more serious. Okay, so these are some of the challenges. We will mostly be addressing the computational challenges in this particular session. The statistical challenges, excuse me, I want to mention, but this is something that we won't explicitly address here. Okay. Okay. So let's try to address these computational challenges. We want to be able to come up with a time and space efficient algorithm for this short read mapping problem, where each of these billions of short reads have to be mapped to a large reference genome. Okay. But haven't we already seen pattern matching in our course, right? A problem like this in our course. So let's just look at if some of the existing algorithms will work for it, right? In fact, even before looking at these existing algorithms, we may want to solve this, right? So let's just put some numbers here. So if our genome is L in length, L is 3 billion letters, right? Then each of our read is m in length, right? And then there are n such reads where n is also in the billions, right? Then what is the naive way of running this, right? So I can write a pseudocode which will say for each read, right, this will go over n reads. This loop will run n times. Scan this read across the whole genome. Report the first hit. Okay, if it hits in multiple places, maybe you want to report it. If it hits in 2 places, you may report that with confidence score half. It maps to this first position, and with confidence score another half, it maps to the second position. Okay, but for now, let's just assume that the reads only hit to one position, or even if it hits to multiple positions, we are always going to report the first hit. Okay, so then for every read, so there are n such reads, right? n is again in the billions. So this outer loop is going to go for n reads, and scanning this read across the whole genome is going to be a loop on its own, right? So for every position, for each position in the genome, there are L such positions. Check if read, for each read I, right? If read I matches for each position j in the genome. Okay, so position j. So you have a position j in the genome, and you have the read I. So you look at j plus 1, etc. to j plus m minus 1. So this is your m length read. You check if the read and the position in the genome, if this is A, etc. to C, you just check if the read is also A, etc. to C, right? Then if that matches, then this is a hit in the genome, right? So essentially this is like a brute force naive algorithm, right? So where we go over all the n reads, and then each that read you just first match in the first position, second position, third position, fourth position. You keep on matching it to this jth position, and j runs from all the way from 1 to L, right? So the inner loop is running from 1 to L, and then you check if the read I matches position j, and that takes another m running time, right? Order of m running time, right? And then if you find a hit, you report it, right? So that is what these two statements map to. So then your running time is going to be outer loop is n times running, inner loop is running L times, and then there is one more loop, inner loop, that will check if the read I, if the read matches at this position j, right? So that is order of NLM. So this is going to be a disaster if you try to implement it in practice, because n is in the billion, L is in the billions, right? And m is again in 200, so this is going to be a billion times 200 base pairs, let's say. So, and with the constant and other factors, this is going to be a very long running program. Okay, and so what we would like to get to is, can we get to prevent this kind of quadratic dependence, especially on n and L? Instead of order of NL, can't we get something like order of n plus L? Is that even possible, right? And linear is good, because anyway to read the input we need to spend at least linear time. So instead of order of NLM, can we do something like order of n plus L, ignoring m factors, right? Can we do something like this, right? So that is the goal, and that is the reason that even though we have seen some simple pattern matching algorithms in this course, we don't have the tools yet to address this problem to come up with an algorithm that runs in linear time. Okay, and I want to show this slide so that you may recall that this is the slide that came up in finding the origins of DNA replication, right? And then there we had this most frequent k-mers problem, right? We were given a string, right? We were given a string and we wanted to find all the most frequent k-mers in a string. And then, so this is a string text and an integer k, all most frequent k-mers in text, and we came up with multiple solutions of varying difficulty. I actually elaborated on this text square times k, which is more like this, the kind of loops that we saw. So it has a quadratic dependence on the size of the text. But then there was another one where we can go over all the k-mers and then scan the k-mers across the text, right, to figure out which is the most frequent k-mers. So the second one is better than the first one if k is not too large, right? So if k is small, so this k-mer is a short string. And then we didn't actually look at these other two solutions, but those were left as exercises. And this text into k-log text may be, you may have figured out it as part of an exercise, but then this linear running time is actually what we could achieve with some of the techniques that we will see in this session today. Okay. And we, in fact, we predicted at that time, or we said at the time that you will later see how a naive and slow algorithm with this quadratic running time, text square times k. In our case here, we call it as order of n times L times m, right? This is just cubic dependence, quadratic in text size and k, can be turned into a fast algorithm that is linear in the size of the text. Okay. And again, like I said, this is how the algorithms or techniques that we will see today will lead us to this linear running time, and this can also be used to solve this finding most frequent k-mers problem. Okay, what we will see today about suffix trees and suffix arrays, those are the techniques that we are going to see. Those suffix trees and suffix arrays and those suffix related data structures will not only solve the short read mapping problem to go from order of NLM to order of n plus m, but it will also make the finding the most frequent k-mers problems that we saw earlier in finding origins of replications of DNA, right, in that session, that can also be solved not in quadratic time but in linear time using the same techniques that we will see today, namely suffix trees or suffix arrays. Let's also look at the sequence alignment. I mean, haven't we already solved the problem of comparing two sequences? Can that be used to solve this problem, right? But in the sequence alignment problems, we were mostly considering the edits that are required and changing one sequence to the other sequence, right? So we wanted to introduce these gaps, these insertions and deletions, and we were allowing some substitutions also, right? Mismatches to change one sequence into another sequence. And we saw this longest common subsequence problem as one way of formulating the problem of comparing or aligning two sequences, right? And we saw how if you put one sequence this way, one sequence goes this way and the other sequence goes this way. So this may be sequence 1, this may be sequence 2, and then we saw that the general alignment problem is a global alignment problem where you want to align the full of sequence 1. This could be A, C, C, G, etc., and this could be A, C, C, G, and so on, right? You want to align the entirety of sequence 1 to the entirety of sequence 2. That is the global alignment problem. And local alignment problem is you just look at the substring of sequence 1, in this case ACC, to the substring of sequence 2, in this case whatever letters is in the sequence 2, right? So you can also form this local alignment problem where you are aligning a substring of one sequence to the substring of another sequence. What do we have in our current short read mapping problem, right? We have one sequence that we have, which is the genome, which is a very, very long string. And then we have a very short read, right? This is your short read. Very short compared to the size of the genome. and we don't want to align the short read to the full genome, right? We are looking for a substring in the genome where the short read would align, and mostly without any we are not going to consider insertions or deletions, and we will consider a few mismatches, but initially when we are presenting the problem, we are only looking for exact matches, right? So then we are essentially looking at this location in the genome where you don't have any insertions or deletions. You start at this top left corner and you come to this bottom right corner. So this is matched in its entirety. The short read is matched in its entirety, right? It is global with respect to the short read, but then local with respect to the long genome. So I call it this global local alignment, if you will. And this has to be done not just for two sequences. The genome is the same, but it has to be done for many short reads. The genome is the same. So this is still the same genome, the reference genome. This is the reference genome. All of these reference genomes in these alignment problems are the same. But this is short read 1, this is short read 2, this is short read 3, this is short read 4, and so on, till millions or billions of short reads, okay? And then this may match here, this may match here, this may match here, this may match here. So this is how this problem of short read mapping relates to the alignment or sequence alignment problem, okay? What we have in terms of short read mapping, right? Even though there has been a lot of work done on string matching and sequence alignments, right, both global and local alignment, short read mapping is still a unique problem where it is a global to local alignment where you are matching the entirety of a set of patterns against a long string, okay? That is what it is in terms of computationally, where the set of patterns is all the short reads, right, set of short strings or patterns, and the long string is going to be a large genome, okay? And you're doing a global to local alignment of the short reads to some substring of the large genome. And like I said, we are not going to consider insertions or deletions. We are only going to permit exact matches and later allow mismatches or mutations, okay? So allowing only exact matches and later allowing mismatches or mutations may seem to be limited, limiting the power of the problem, but it will turn out to be incredibly useful for finding disease mutations, because even with this simplification, we can find disease-causing mutations. Many of the mutations are not insertions or deletions, they are single point mutations. So by just considering exact matches initially and later allowing mismatches, we will be able to find these mutations. So it is very practical in the context of finding disease mutations. And at the same time, it's also very fruitful because we have made these simplifications where you have a set of patterns and you have a long string and you want to match each of these patterns and find the location in the string where these patterns occur, right? As a substring, we have simplified the problem computationally so that we can find efficient algorithms, okay? And that is what we will be looking at a bit more carefully in the rest of the slides, okay?